welcome back to no. Actually, no. Hello, everybody, and welcome to BioQuizzical. This is another podcast, vlog, thingamajig, whatever you want to call it. So this is another podcast, and what we're trying to do for Biology Week is to do a podcast every day for the 5 weekdays on biotechnology, and trying to get maybe the community involved as well, and sort of speak about things in a way that anybody can understand. And today's topic we've chosen is biosensors, because they're really important and they're really, really cool little devices usually, and they so they're really cool devices, they're really interesting, and they're becoming more and more prevalent, aren't they? Especially in things like medicine and stuff like that.
Yeah, so this has been a bit of an eye-opener for me in the last few days. I've sort of basically started researching the topic from fresh, not knowing a lot about them. But there seems to be quite a wide variety actually in these things. They seem to have been around for a little bit longer than I anticipated, because when you hear the word biosensor, you would think it's a fairly recent thing, but these actually, the research and the development of these things actually dates back to about the 1960s.
Yes.
So it's been a thing in the pipeline that's obviously just gone absolutely crazy in the last 2 years. And we were talking just before the podcast of what were the figures you mentioned about the value of the industry?
Well, by 2022, they reckon the value of the industry will be just a little bit over $27 billion, and then by 2024, maybe $29 billion. And if it's in dollars, that probably just means the US.
Yeah.
So that's not worldwide either.
It's crazy. Absolutely massive.
Yeah, and if you think about it, biosensors have been around for a while. I think we're all very familiar with the blood sugar test for diabetes. That you just put a drop of blood on it, and it just tells you what your blood sugar is. Brilliant biosensor. Pregnancy test is another biosensor. Pee on a stick, tell you if you're pregnant.
Do you know the old way of doing it?
Taste test?
No, they used to inject it into a frog. They used to inject women's urine into frogs, and then if I remember rightly, they used to change.
They changed sex.
If they changed sex, then it meant that there was like a certain level of hormones, so therefore that was a legit pregnancy test at one stage.
Can you imagine getting that in the mail? I said dead frog again. Not pregnant.
Yeah, so I think it's worthwhile mentioning that there's a difference between a biomarker and a biosensor.
Yeah.
So a biomarker being a detectable compound or molecule within a solution, so whether it be blood or tissue fluid, that would indicate a certain thing. So you might find a certain isoform of enzyme in blood, so your heart isoform in your blood would detect that you've had a recent heart attack.
Yeah.
So that would be a biomarker, whereas a biosensor would be, would you like to give an explanation?
Well, a biosensor is basically a device that uses usually a biological organism or something that comes from a biological organism to detect a biomarker or something like that.
Or some sort of animal.
Some sort of, so basically the basic biosensor is you have a receptor for a very specific analyte that you're looking for or you're trying to detect, and if you do detect it, that goes on to trigger a transducer, and that gives you some sort of electrical or visual or some sort of output a quantifiable output as well, and something that you can measure. And that could literally just be, we're very familiar with how fireflies light up using an enzyme called luciferase, and we're very familiar with the fact that some of these biosensors just use luciferase, so if you get positive results, they glow. And it's nice and easy, and it's really quick, and you can use them out in the field. And they use them for things like herbicide detection in water systems.
Yeah. So, and biosensors are a major part of synthetic biology, aren't they?
Yes.
So it's an emerging area of science that's come about in the last sort of 10 to 20 years, really.
Which is why a lot of these biosensors are only just getting out there, because it takes so many years to test and to get approvals and then to use them commercially.
Yeah, and traditionally a lot of the way these things have been used is by using bacterial systems.
Yeah.
Or systems of other microorganisms, which aren't necessarily, there are biosafety issues there that you have to be able to be aware
So they may not be dangerous to our health, but you don't want transgenic bacteria getting out into the environment.
It's a very similar ethical argument to the GMO argument.
It's pretty much the same.
So, I mean, so when you look at a lot of these biosensors nowadays, they have inbuilt kill switches, don't they? So basically, once the bacteria get within over a certain level or over a certain age, they'll actually initiate a cellular program which just causes death inducible into them. So we got some examples of some biosensors that we wanted to discuss. That was part of.
Yeah, definitely. I know that there's one at the moment. I think it's still in testing, but I read it on an article. Sorry, I can't remember where from. Maybe PLOS One. I'm not sure though. Yeah. But anyway, they want to be able to detect cancer because the thing about especially medicine is the faster you can detect something, the more chance you got to cure it or at least treat it. So they want to detect cancer really, really quickly. So what they use is a chip. It's contoured in a way that these big cancer cells, they won't. It basically uses the physics of how they flow, and because they won't flow as well as the rest of the blood cells, that they can be detected on outside detectors, and basically they can detect if there's cancer cells just by. And the chip's only like that big, and they just wash the blood through it basically, or any sample you want through it. And you can do that, you can repeat it, and it takes minutes. You know? So
they'll be able to basically diagnose cancers at early stages rather than having to wait and obviously increasing mortality rates.
Yes, you know, the earlier you can catch cancer, the better you can treat it.
Absolutely. Yeah. Well, I read an interesting one that actually the paper dated back to sort of the early 2000s, sort of pre-2010 certainly, about when they'd actually engineered bacteria to be able to, if you ingested them in, let's say, a tablet form, they would actually move through your bloodstream, or if you injected them either, probably slightly better, they would move through your bloodstream and move towards a cancer cell, and they would actually invade the cancer cells, and they'll only start to grow once they're inside, because tumours typically have a very low, so they run on glucose alone. Because they're running without oxygen, effectively, the anaerobic environment. So that you generate these bacteria that are anaerobic, that thrive in these low oxygen areas, like inside a cancer. And then what happens is, once they're there, they turn on a system which generates anti-cancer drugs, and then once they get to a certain population size within the cancer, they will turn on the same kill switches, will just basically burst the cells and dose the cancer from the inside out with these anti-cancer drugs. It's far more targeted than a traditional chemotherapy.
Well, I was reading about a proposed biosensor on the iGEM. So iGEM, if you don't know, is the International Genetically Engineered something competition.
Pass.
I
just know it's iGEM.
It's iGEM.org. It's really, really interesting. It's a competition, and basically, all these people they submit, you win awards for the way a competition works. But one of them was the fact that, like you said, it's targeted cancer treatment where they take an organism they've genetically engineered, and what happens is cancer gives off very specific drugs and stuff like that, and it makes very, very specific molecules as well. This works off two of them, basically, so it's kind of got a little bit of a fail-safe there. You need both of these, and when both of these chemicals are detected, they transcribe guide RNA and also another molecule. I wrote about this like 2 days ago. And another molecule. Basically, both of these, once they're transcribed, they conform with each other. They associate with each other, and then they drive transcription of apoptotic initiators and things like that. So they basically just, rather than pumping your cells full of chemicals or anything like that, they target cancer cells and cause cancer cells to kill themselves, effectively. But these can be reprogrammed to also inject fluorescent proteins into tumours instead of killing them. So if they need to be surgically removed, the surgeon can easily see where all the tumour cells are.
Oh, that's interesting.
And they also are programmed to even just locate cancer cells in that way. The only drawback on that is you have to personalise the organism for each person you're putting it in, because as you may or may not know, that every cancer is different. That's why you can't get an overarching cure for cancer, because every cancer is different. So probably your DNA.
I think it's a common misconception that the actual definition for cancer is any cell which is dividing uncontrollably.
Yes.
Whereas everybody has this certain idea. So there's many cancers as there are tissues in your body, and many tissues in your body there are multiple cancers as well.
And because it's a problem
for different people.
Yeah, exactly. Because it's a problem with your DNAs.
Yeah.
Each cancer can almost be treated as a unique case.
Absolutely.
Even though you can get some overarching treatments for each little section of cancer, it's really, really difficult.
But it's the holy grail of medicine at the moment seems to be this move towards personalised medicine.
Yeah.
Using your genetic code, which is in the sci-fi movie is brilliant, but right now I just don't think we're there yet. We haven't got the ability.
We don't understand. And there's also big chunks of DNA that we don't understand what they do.
Yeah, I think there's a massive misunderstanding about what's achievable. But not only that, is in order to do this, you've got to have your full genome sequenced. And then on top of that, there are ethical issues around the idea of who holds the information in your genome.
I was about to say that people are very uncertain about even giving up their genetic information, and because there's been a bit of hysteria around that.
I've got friends who don't like giving me their bank card, let alone their whole genetic sequence.
I don't like telling people my full name.
But so obviously medical therapeutics is one sort of angle of biosensors, but another one that I've seen a lot of papers around is energy production of fuel production as well. So using hijacking bacteria to then basically turn basically unusable molecules that hijack their amino acid metabolism in order to generate biofuels.
Yeah.
Which is one way of detecting, or one way of basically reversing our effect on the environment.
So they make these biosensors so you can go into the field, like the environmental biosensors, so you can go into the field and you can test waterways there and there. And as we were talking about making things just faster and easier and streamlining things, you can take a little biosensor or whatever, and you can put it in water, and you'll be like, oh, there's a contaminant, I'll take a sample and find out what contaminants there are, because these biosensors, they detect multiple ones, and they just give you a reading. But you could, like I said, you could take 100 samples of everything around you, and 90 of them not have contaminants, and what a waste of time, money, and resources it is to test all of them if you could just, stick these electrodes in and go, no, that's fine.
And also a big part of what we're doing with these biosensors is actually reducing the need for the big lab equipment and being able to basically make this a portable system which gives you much faster results than what you typically get if you have to bring all, like you said, all those analytes back to the lab.
Yeah, because you can't take the lab equipment with you, so you've got to bring hundreds and thousands of samples of soil and water and everything back with you, and you can cut that carriage down a lot.
So like the small one that I saw on the fruit.
Yeah.
So that's like a tiny little sticker like you have on a fruit that they've inserted a gene from coral that turns blue. They've made it turn blue after it senses the thylline when the fruit is ripe.
Oh yeah.
So then you can pick out the right fruit to put on the shelf or out or.
Which is great because you don't have to do a lot of modification in order to get that from an idea that you produced in a lab to a commercial product.
And it's tiny.
Yeah, exactly. So that wouldn't. Yeah, exactly.
Unaffordable, probably.
If you make it cheap enough that you can just throw it away like a sticker on an apple.
Or the tag for the whole bag, which.
Exactly.
Tray of fruit.
And we always, I think the public always wonder, what's the point? You just pick up an apple and you can just see if it's ripe or not. But like we were saying, I worked as a head of fruit and veg department.
Yeah.
And all you're doing all day is just taking bad fruit off the shelf because it goes off really quickly. But if you could just cherry pick the ones that are ripe for the day, people would come in, they wouldn't have to worry about looking at it because they'd just be like, you can see it's blue, or you can just be like, well, everything's going to be ripe because it's been put out that way. And hopefully then you won't get as much waste because waste causes, in landfill you get methane, carbon dioxide, which is environmental problems again, and that's other environmental problems caused by
food wastage. And they could probably
wastage.
alter the same technology so that it works for other food.
Yeah, exactly.
Not
I'm
necessarily ethylene, but like other, I don't know, what.
I mean, you can see it with meat because obviously when meat starts to go off, it does start to generate compounds that you can smell. So
and you can go like turn slightly green.
Or like dairy products, you can probably do it with that as well. Like
as long as they don't make the mistake of putting a number on it, like, so you don't end up with an actual use by date. Because as soon as you end up with a use by date, people will go, well, no, that one's going off on the 15th. Oh, this one's going off on the 17th.
Oh, use by dates are a whole other problem.
Yeah, exactly.
But if it was just something that went blue when it was off, you wouldn't have to.
I like milk about dairy. Actually, dairy is really interesting because there is a transition period where dairy is, it's not great for you, but at the same time, it's not going to like make you. It's not going to put you on the toilet all day, is it? But I mean, I've had milk that's like on the edge, and it's tasted fine.
Yeah.
But afterwards, I've not felt too great, and it's just like, if there was like a sticker or something, or a stick that you just put it in and be like, that's starting to get discoloured. Maybe this is off.
Yeah, but another thing with the dairy thing was that obviously there's a lot of talk from the vegan community about the basically eating dairy and things. One of the big outcries that they have is the use of antibiotics in the dairy industry.
Yeah.
Now again, one of these biosensors that I've seen is it's basically like I say, almost like a stick, so they can put test on the large vat that comes straight out of the dairy, out of the actual parlour, to test for antibiotic.
And you can
presence within the milk. Yeah, so you can test an entire batch for antibiotics. So a big concern as part of the dairy industry is that they use a lot of antibiotics in order to maintain health of the animal. In the cow, yeah. But then they're saying that if those antibiotics then get into the milk, you're then introducing antibiotics into the environment. I read a statistic the other day, which is about 70% of all antibiotics used end up in the environment.
Because I saw something like that, it was about antibiotics, and there was one they had in hospital that they never used because it was like their backup of But then they found that it was being used in animals, like sheep.
And just getting into the environment.
And they use it on all the animals, not just the ones that are ill, so we're using it anyway.
I would say, so bringing it back to, because biosensors, and if you don't know the reason why we use antibiotics and biosensors, is as a marker to show that whatever gene we're trying to put into, say, a bacteria, has been incorporated. Because if the gene goes in, so does antibiotic resistance for a specific antibiotic. Like, it's always usually ampicillin.
Yeah.
So you can stick that on an ampicillin plate and the bacteria won't die. That's great, because you know your gene's gone in, and that's the way. So that's why they use antibiotic resistance.
Because they both function together. Like if that
one's
gene's
got in, both
the
got
other
in.
one's work.
Also, I said like I recounted a story of basically putting in an ampicillin resistance vector into a bacteria to a friend of mine who isn't in biology, is an educated individual, but not in biology, doesn't is not in it as much as we are. And the face of horror that he gave me is if I was going to create the next I was going to create a zombie apocalypse because I'm creating these all these antibiotic resistant bacteria that I'm just going to introduce into the environment, which is just not the way it is.
It doesn't, and to be honest with you, there's such strict controls. In fact, a lot of people quote that it's overly strict controls.
Yeah.
Even though you need strict controls, it's almost a little bit overboard because we are so conscious of that happening.
But that's why they're looking at putting biosensors into yeast rather than bacteria, no? Is that one of the reasons?
Well, that's part of the reasons is that, because they don't pose such a health threat, I suppose. Because obviously with bacteria, you get horizontal gene transfer where they're able to actually transfer pieces of DNA between individuals. That's like me getting you some DNA, which is eukaryotes and yeast, as far as I understand it, can't actually produce.
I assume they're very similar because we have eukaryotic cells, mammals and animals, and so what we do is what's called vertical gene transfer, where we only give our genes to our progeny. Whereas bacteria, like you said, they can actually leach their genes into their surrounding environments, and other bacteria can just come along and sort of hoover them up. And
which is why antibiotic resistance is such a huge problem.
Like walking around, that's a good gene.
There's there's just like, I'll have that. Like that. And then eventually one of those is antibiotic resistance, and then they pass that on to all the cells that they proliferate into, and all the other cells around them.
Yeah, because it's an exponential growth thing, isn't it, with bacteria always. But and again, biosensors are being used in order to create or even certainly detect new antibiotics that they're using. So it's a way of generating these new novel drugs. in order to so that we can actually bolster our arsenal against antibiotics. Because if it was there's something by 2020 is going to be the rate of deaths due to biotic infections that should have been curable with antibiotics which are no longer because of this global problem of antibiotic resistance.
And you think about the history of humans, we've only had antibiotics for less than 100 years. They were found 89 years ago.
And we used them up already.
And in that time,
yes.
So yeah, and that's also kind of highlights how. It's a bit off topic, but how prolific infection is, and going back to biosensors again, this is why we're moving away from what we call whole cell biosensors, where we use these genetically modified organisms, and instead extracting things like enzymes from them, fix them to a thing like paper or
so this is what you call a cell-free system. So you basically take the components that you would use for detecting the analyte within the cell, and rather than having it within the cell, just have it simply fixed into whatever it is. You mentioned paper as like a litmus test, but obviously the big one at the moment is graphene.
Yes.
And with combination with material science.
Yes, yeah. And they're testing new ones, I think. Gold nanoparticles are used in there as well.
That sounds so sci-fi though.
I know, it's amazing.
Just nanoparticles, and I'm like...
The fact that I could get in the word nanoparticle pretty correctly.
Yeah. So what are they doing with gold nanoparticles then?
Oh, well... but I don't feel so smart.
The ability of graphene, obviously, one of the great things about graphene is it's so conductive, so you can then actually use cellular components as parts of genuine circuits.
Yes.
So rather than thinking about these systems as two separate things, where you've got biological ways of looking at solving a problem, you've actually got electrical ways. Actually augmenting the two together, so that you end up with biological components being a part of a circuit, which is very sci-fi.
They call it like chemoelectrical circuits and stuff like this. And I was discussing the fact that that is basically replacing the whole cell system that we use at the moment with photosynthetic cyanobacteria. And that is because in photosynthesis it works on moving electrons, and you move an electron, you get a small amount of charge. And that means you can put a small amount of electricity through this bacteria, this cyanobacteria, and get a constant ampage. But things will block photosynthesis, pollutants will block photosynthesis and make the ampage go down. And that's how you can use a biosensor in the field. You can put it in a water system and go, oh, the ampage has gone down, there's a pollutant here, it's probably one of so many, I will take a sample. Like I said, you can go around and be like, oh no, there's no pollutant here, I don't need to take a sample.
So would that be similar to what I was looking at with the ion channel, where it blocks the passage of the ions through?
Yeah, if you've got like a voltage gated ion channel.
And they've made a biosensor which can light up depending on voltage.
But what we really want to do is just take the binding site effectively, put it into like an enzyme or something like that.
Which is great theoretically, but of course it's very complicated. As much as you like to think of enzymes as Pac-Man, just this idea of it's just been chomp chomp chomp. The 3D structure actually determines the function of the binding site. Sometimes you won't get correct shaping of the binding site, correct protein folding.
And if you change the binding site, you change how the protein might fold.
Absolutely.
And then it might not bind to your promoter site to promote your signaling gene, and the whole system doesn't work.
This is why researchers work in teams and not individuals.
Yes.
So part of the way this came about was when they stopped looking at organisms as organisms, and then they started looking at them as circuitry. So if you're trying to create a circuit, making a light bulb go on, you might put a resistor in there, you might put a switch in there, and you get an outcome of a light bulb. But if you change the idea of resistor and switches and light bulbs to enzymes or luciferase proteins or even any of these genes themselves. And basically take that idea, you essentially got what they're using biosensors. It's very simplified, but actually they actually work.
But it does go back again to what we said about biology. It is simple. It's at least the idea of it. We've been discussing our own biosensors this week, and we've been saying, oh well, I want this to do this, and then you think, okay, well I'm going to need this part. Oh well, if I need that part, I need this promoter. Oh, if I have that promoter, then I need this, and it comes together very easily. And obviously, there's a level of like education there as well. But the idea of it is nice and easy.
The complicated bit is understanding the enzyme. Like I found, I wanted
the
enzyme, but they didn't know what the binding site was.
Yeah, the complicated bit of it is if you want to make something really new.
Yes.
And like with you, there was an enzyme that had been found, but no one's ever sequenced it, and no one knows its binding site.
There was one where they had the main binding sites, but not all of them, and the one I needed was not one of the main ones.
Yes, exactly. You know.
But it's very easy to explain in layman's terms what these systems are doing, but actually the technicalities of what they're doing is actually extremely complicated.
Which is why it's a huge business.
Yeah.
You know.
Yeah. So because it was, we were looking at, so I wanted to achieve a certain sensor yesterday, and it took 3 or 4 of us to actually come up with a way of going, actually, well, rather than doing it the direct way, which is the most logical, well, if I turned it around and went this way, it would work a lot easier, utilising different biobricks that other people have used.
It took 3 or 4 of us, including an expert, an hour and a half to figure out, we might be able to do it.
Saying for me that I probably couldn't do it.
It was when we were talking about it, and then we were reeling off the list of things we had to do, and we were kind of like, so it's a bit overkill, really, for what we need to achieve.
Yeah,
so,
for the timescale that we've got, this is extremely complicated, and that's another reason why it takes so long.
Yeah.
You know, these,
so a good iGEM competition project would take at least 20 weeks worth of wet lab work, let alone dry lab work and all the modelling and the bioinformatics work on top of it.
Yeah.
So while these things are great and there are a massive emerging technology, the work behind them is really.
I certainly think that within the next 10 to 20 years, there's going to be so many more biosensors. Even if it comes down to commercial like fruit ripening.
And I don't think the word biosensor will enter the public sort of.
They'll make it snappy.
Vocabulary. Well, not only that, I just don't think it'll enter it because we think that one of the first things you start talking about as a biosensor was a pregnancy test.
Yeah.
You don't think of that as a biosensor. You think it's a pregnancy test, and I think it will enter you. It'll be one of these things that
it'll be there, but you won't know that.
It'll be a nifty gadget.
Exactly.
Yeah, because I don't think I knew. Like, obviously, before we started talking about biosensors, I wouldn't have thought, pregnancy test.
No, exactly. That's the great thing why I enjoy doing this as well, because you don't realise, you sit down and think about it, actually these things have been around for decades.
Yeah. Is that worthwhile holding it there then?
Yeah, I think so. I think it's just a very interesting topic, and I think it's something that people are going to see in the market more and more once they get approved.
Absolutely. So that was podcast number 3, I believe.
If they come out in order.
If they come out in order.
If not, it could be anything.
It could be anything. So this will be on our.
They'll all be out in 1 week.
Yeah. So these are going to be released every day this week for Biology Week. This will be released onto the YouTube channel, which is our BioQuizzical channel. Also follow us at BioQuizzical on Twitter.
And Biotech Musings.
And Biotech Musings for Maddie as well. Hit the subscribe button if you like what we're doing. We're trying to basically bring down these large concepts into something that's a little bit more manageable. And we should be doing a few more of these.
Yeah, and I would like to say that other than other workloads that I've got, after we finish this podcast, I'm seriously thinking about doing little snippet videos about interesting topics, and I might diverge away from biotechnology a little bit. So this YouTube channel, it's not really just been set up for Biology Week this week. It could be a continuing thing, and it could be worth hitting that subscribe button if you want to hear regular people badly explain biology that no one really understands.
But and we'd like to hear back from you if you're interested in it. If you want us to hear us talking about a certain topic or maybe explaining something.
Suggest a topic.
You've heard in the news that you're not sure of. Because there are, it's becoming bigger and bigger news every week.
And I've noticed the news are getting more technical with what they report, and that can be quite
daunting.
Yeah, it can for the regular reader.
Yeah. Cool. So look forward to seeing you again.
Well, thank you.